So now let us start with the introduction. What is virus? Actually, the name virus stems from the concept of a poison or illness that appears to move in the air. According to Kahn, they are submicroscopic, obligate intracellular parasites which reproduce inside their host cell. They are too small to be seen by the optical microscope, but they are composed of a genetic material either by DNA or RNA. They are considered to be organic structures and they interact with other living organisms. However, unlike any other life forms, viruses do not have their own cellular structure, they do not have their own metabolism, and they cannot reproduce outside their host cell. That's why, by a strict definition, virus cannot be considered a living organism. There are also different types of virus which infect different host cells. There are viruses that infect plants, humans, and other mammals. Both prokaryotes and eukaryotes can be infected by viruses. We will only focus on animal viruses. So there are different families of animal viruses which cause different illness and diseases. The first genome category is the dsDNA genome, which includes the family Poxviridae, the largest and most complex among viruses, family Herpesviridae, which establish latent infection, family Adenoviridae, which is first isolated in human adenoids, family Papillomaviridae that are resistant to low pH, and family Polyomaviridae that causes wart lesions to humans. The second category is the ssDNA genome, which includes the family Parvoviridae that causes arthritis and erythema. The third category is the dsRNA genome with the family Rheoviridae, which causes non-bloody diarrhea and Colorado tick fever. The fourth category is the positive ssRNA genome, which includes the family Coronaviridae, the primary cause of SARS or severe acute respiratory syndrome, family Picornaviridae, which cause hepatitis and foot and mouth disease, family Flaviviridae, the main virus causing dengue, family Togaviridae, family Astroviridae, and family Caliciviridae. The fifth and last category is the negative ssRNA genome. It includes family Paraxoviridae, family Rhabdoviridae, family Filoviridae, family Orthomyxoviridae, and family Arinaviridae. It is actually important to have a profound understanding of these viruses because through this, we can see how intimately they are connected to ourselves.
The origin of viruses was being debated among virologists, even though they have accumulated a significant amount of knowledge and information about how the viruses now evolve in the present. Viruses do not fossilize, thus they did not remain traces of evolution in the past. With this, researchers can only come up with hypotheses through investigating how the viruses we see today evolve and through the use of biochemical and genetic information gathered. There are three major hypotheses which have been articulated according to Wessner. First is the progressive or escape hypothesis, which states that viruses gain the ability to move in between cells through genetic elements exhibiting this ability. This hypothesis suggests that viruses originated from RNA and DNA molecules that escape from a host cell. Second is the regressive or reduction hypothesis, which states that viruses are remnants of cellular organisms. This is also called the devolution hypothesis, which proposes to explain the origin of viruses by suggesting that they evolved from free living cells. Lastly is the virus first hypothesis or the self-replication hypothesis, in which viruses predate or co-evolve with their current cellular host. Several studies of plant pathogens support this third hypothesis. Since we only have the present-day viruses to observe and determine their evolutionary steps, scientists found the mechanisms of virus evolution through mutation, recombination, and reassortment. A combination of these molecular events, according to Domingo, would produce a complex and phenotypically diverse populations of viruses. The molecular basis for high mutation rates is the absence of a proofreading repair activity. With this, mutation is not an occasional event, but a continuously occurring event, meaning viruses would then have the chance and ability to evolve rapidly. The next mechanism would be recombination, which occurs both with RNA and DNA viruses. This has been viewed as a means to rescue fit viral genomes from low fitness parents or a means to produce highly divergent genomes, providing opportunity to explore the adaptive potential of rare and new genetic combinations. Lastly, genome segment reassortment is a major evolutionary force for some viruses with a segmented genome.
For our paper, we will be focusing on RNA viruses. So their characteristics is that their genome is either single-stranded or double-stranded. Their genetic material may either be monopartite or multipartite, though it is more common for them to have multipartite genomes than it is for DNA viruses. Single-stranded RNA viruses are further divided depending on the material they use for encoding of genes. As they are divided as positive ssRNA or negative ssRNA. They also have smaller genomes than DNA viruses, which code for 1 to 13 proteins in general. And because of that, replication occurs rapidly for them, causing mutation to occur more likely, and as a consequence, RNA viruses evolve faster. The molecular evolution of influenza viruses will be based from the study of Suzuki and Maie. So the evolution of HA genes is given emphasis on to assess the evolution of influenza viruses, as influenza A virus pandemics arise upon the presence of new HA gene subtypes in aquatic birds, which is the natural reservoir of the virus. HA genes of influenza A and B viruses diverge prior to the divergence among subtypes of influenza A virus HA genes. In the natural reservoirs, there is a rate of 10 to the negative 4 amino acid substitution per site per year, by which HAs and HEs in the case of influenza C genes evolve. Serotypes in humans are based from the viral proteins hemagglutinin and neuraminidase. There are 15 serologically distinct HA proteins and 9 for NA proteins, which are named chronologically as HA, H1, H2, and so on, and N1, N2, and so on. From these, the serotypes infecting humans are the combination of H1, H2, and H3 with N1 and N2. The differences in the biochemical functions between the viral proteins HAs and HEs and the natural reservoirs among influenza A, B, and C viruses allowed such divergences to occur. As early as 412 BC, a record on human influenza pandemics was found. Thus, the study of Suzuki and Maie suggests that more than 2,400 years ago, influenza A virus has existed.
Family Picornaviridae. Members of family Picornaviridae have RNA molecules ranging from 7.2 to 8.5 kilobases in size. Its infectious genome consists of positive single-stranded RNA with non-enveloped icosahedral cups. Consists of densely packed icosahedral arrangement of 60 protomers. Each protomer consists of 4 polypeptides, etoposide 1, 2, 3, and 4. The capsid coat protein serves multiple functions. Epidemiology. Picornaviruses are transmitted most commonly by the fecal-oral route or respiratory route depending on the specific virus. They undergo the process of replication that happens in the cytoplasm. The poliovirus responsible for poliomyelitis among humans is considered to be highly infectious. Serotypes in humans. For enterovirus, there are more than 60 known serotypes wherein the primary replication of the virus which enters the body via the alimentary canal takes place in the pharynx and gastrointestinal tract, and the virus can be found in throat and feces after the viral shedding. Manifestations can be divided into 3 categories. Number 1, abortive poliomyelitis. Number 2, aseptic meningitis. And number 3, paralytic poliomyelitis.
Poliomyelitis can be prevented by a vaccination, and there is no effective antiviral therapy available for it. Next is rhinovirus. Rhinovirus currently has 103 different serotypes and is transmitted via aerosol droplets, virus-contaminated respiratory secretions. It is the most frequent cause of common colds. Hepatovirus is responsible for a self-limiting viral hepatitis in human beings and may be transmitted by the fecal-oral route during acute infection or by the ingestion of uncooked contaminated shellfish. There is only one serotype of hepatitis A virus and is highly contagious. It can be transmitted through fecal-oral, sharing of needle, blood transfusion, and poor sanitation. There are several laboratory tests to undergo to confirm the onset of hepatovirus. Last is the aptovirus. Aptovirus is the cause of major global animal health problem. Humans interacting with infected animals and who inhale droplets of virus experience symptoms like fever and vesiculation on skin and mucous membrane. Other possible methods of transmission are ingestion of infected food and contaminated semen. For molecular evolution, it is classified on the basis of physical characteristics and antigenic relationships measured by neutralization tests. Genome diversity are organization patterns and common genome features based on the analysis of mammal-infecting precoronaviruses, the ordering of intracellular membranes, and number of potential mechanisms for RNA virus vesicogenesis for driving membrane formation and lipid synthesis required for viral replication is known.
Family Coronaviridae is a monophyletic cluster of the order Nindiviralis. They are classified as envelope-positive strand RNA viruses with non-segmented genomes of about 30,000 nucleotides. The members are known viruses of mammals, birds, and even fish. Coronaviridae is comprised of two subfamilies, the Coronavirinae along with Torovirinae. The Coronavirinae is further classified into alpha, beta, gamma, and delta coronaviruses, which used to be sorted based on serology but are now divided by phylogenetic clustering. Coronaviruses and toroviruses are distinguished by features such as the size of the nuclear capsid protein and the shape of the helical nuclear capsid structures, as well as gene products and the presence or absence of leader sequence at the end of mRNAs. Coronavirus is derived from the solar corona-like appearance of virus particles in negatively stained electron micrographs, while the name torovirus came from the curved tubular morphology of its nucleocapsid structure. As of present time, 13 viruses are classified as species under the family Coronaviridae. Given the properties of coronaviruses, its variants are said to be sensitive to heat, lipid solvents, non-ionic detergents, formaldehyde, oxidizing agents, and UV irradiation. Coronaviridae generally possess 3 to 5 structural proteins, namely spike membrane envelope glycoproteins, hemagglutinin esterase, and nuclear capsid protein, all encoded within the genome's 3' end. According to family, it is important to note that the function of most of the structural and accessory proteins encoded by these viruses still remain unknown up to date. S proteins of the coronaviruses mediate the entry of the virus into host cells. The M protein, on the other hand, plays a role in the diffusion of the virion into the cell and its regeneration inside it. The E protein facilitates the assembly and development of virions within the cell. And lastly, the N protein has a vital role in terms of the virion structure, replication, and transcription of coronaviruses. Details regarding the replication of coronaviruses remain largely unknown. This is because these viruses are difficult to grow in the laboratory. The mouse hepatitis virus is currently the model being used for the replication of coronavirus. Replication starts with the attachment mediated by the spike proteins. Human coronaviruses enter cells by specific receptors such as aminopeptidase N and sialic acid containing receptor. Genomes are released into the target cell through the fusion of the viral envelope with the plasma membrane or the limiting membrane of the endocytic vesicle. This entire cycle of replication takes place in the cell cytoplasm, wherein a full-length and subgenome-sized minus-strand RNA intermediates are produced for each viral polypeptide. This minus-strand RNA serves both as the mRNA for replicase proteins and as a template of the minus-strand synthesis. The genomic RNA then binds with the nucleocapsid progeny virus derived from the endoplasmic reticulum and Golgi apparatus are then packaged into vesicles. Said vesicles travel and fuse with plasma membrane, followed by the release of viral particles in the cell. Coronaviruses are highly species-specific and spread from an infected person to others through inhalation, direct personal contact, indirect contact, and even fecal contaminations. Since the virus replicates locally in the cells of ciliated epithelium, cell damage and inflammation are inflicted on the host. People can get infected any time of the year, with young children most likely to get infected. Not until the discovery of SARS-CoV in 2003, there was only 2 coronaviruses known to affect humans. These 2 are human coronavirus 229E and OC43, first described in the 1960s from the nasal cavities of patients with a common cold. Moreover, the next discoveries of another human coronavirus was in 2004 and 2012, HCOV, NL63, and human coronavirus HKU1 respectively, and in an outbreak in 2015 is the discovery of the 6th human coronavirus, the MERS-CoV. Coronaviruses have repeatedly emerged during the past 1,000 years. The 61 coronaviruses belong to different lineages and are closely related to animal viruses, with most originating from bats and are transmitted to humans either directly or indirectly. This is implying that coronaviruses have propensities for host jumping. It is thought that some of the human coronavirus accessory proteins were acquired from host genes, and some were lost or were split during its evolution.
It has been more than a century since the first discovery of virus in 1890s by 2 scientists, Ivanovsky and Bacod. Although there had been lots of discoveries about viruses in the recent years compared to the earlier times, we are sure that there are still lots of undiscovered viruses in the world. Virus discoveries may be caused by easier diagnostic due to technology's development, or it could have been the changes in poultry farming and animal management practices, changes in climate, and other ecological factors. However, the rate of change of these factors is unmatchable to the rate of increase of discovery of viruses. Now, this suggests that we should look into other factors that could have influenced the emergence of virus. One of the products of advanced technology is genetic engineering. Genetic engineering allows the editing of genome of any organism, including virus. Therefore, this means that laboratory engineered flu is possible. Now, here are some measures to prevent pandemic caused by viruses. First is the use of pharmaceutical drugs and vaccines, protective equipments like masks, and non-pharmaceutical intervention like quarantines. Provided with advanced technology, it is suggested that researchers look into both the virus and the host's perspective. This is for us to be able to see clearly on how these two interact and how each adapt to its environment. There are still lots of studies needed to be conducted to be able to know how did they originate, what is their function, how do they function, and what are their other properties, and what are the factors that attributes to their emergence, and many, many, many more. And the only concrete conclusion that we can give is that viruses, before its discovery and until this day, continue to modify and adapt to its changing environment.